Welcome back. Now let us look at sample matrix and inorganic analytes. You may recall, sample matrix refers to the holder of the unknown that we are interested to study. And that unknown is called the analyte. So inorganic analytes refers to the inorganic elements in the periodic table. And today we are going to look at sample preparation involved in a biosynthetic sample requiring you to extract out the inorganic analyte. So let us recall it. When we talk about an unknown, we have to have in mind that an unknown contains matrix and the analyte, or the focus of our investigation. So let us think of an analyte as this, having a drum of particles. The analytes are the darker blue and the matrix is made of the light blue background. So to get the analytes out requires sample preparation. The D statement on vanilla distilled water is an example is based on the visit that your seniors made last week for analysis of UV visible spectrophotometer at a food lab. But for your focus, let us just today look at analytes and matrix. So analytes are normally the minority components in an unknown. So we are going to look at how to extract the inorganic analytes. Say we have tin, an analyte, in a protein matrix. This should serve as a good example. What does it look like? What does the sample look like? If you want to imagine one biosynthetic sample, you can take cockles as an example. So, how do we transform this cockle from an analyzable form, liquid form of sample? Of course, in order to get tin out, one way is to destroy the protein matrix. Okay, so let us look at how it is possible to destroy a protein sample matrix. Recall here, materials are made of two possible principal components, organics and inorganics. For biosynthetic samples, the inorganics and organics are hardly pure and they normally coexist. That's what this means. So, since we are extracting the organics out of our sample matrix, looks like we are going to take this route of procedure. So, what this means is, if we are interested to get an inorganic analyte out, okay, we are going to extract the sample. Of course, before extraction, we need to resize, grind to acceptable size for good surface area to release the analytes. Alright, then we do combustion. If you can recall, last week we covered that in order for a complete combustion to happen, to release carbon dioxide and destroy the organic matrix, we need the sample to be small enough so that 550 Celsius degrees plus minus 25 Celsius degrees would allow complete combustion, release of carbon dioxide, and give us inorganic residue. After getting the inorganic residue, we can dissolve it, and there we have it, inorganic sample. So we start off inorganic extraction by combustion, which is a classical analysis. Grind the sample, imagine cockles, okay, dry and grind the cockles and put it in a crucible. Of course, not this type of crucible, not glass crucible, but porcelain crucible that can withstand hundreds of Celsius degrees. Okay, put our sample in, and there we go, combustion at 575. Okay, take the initial weight of sample plus crucible, say 31.0386. After combustion, take another weight, say the final weight is 30.2586 grams. So the loss actually represents the organics released or destroyed during combustion, which is carbon dioxide. So when we minus 31.0386 with 30.2586, the residue represent the inorganics contained in the sample. So the ash is the residue, so that weight is actually your ash. So percentage ash is whatever residue divided by the initial weight of the cockles times 100. So that's percentage ash. So we begin with gravimetric analysis and obtain gravimetric data, which is percentage ash. So that's one method of extracting out the inorganic analyte. Another way is liquefaction. If we have a specific digestion unit, we add acid to the sample, to the cockles for instance, let it dissolve in concentrated acid, we call it digestion, and then dilute the sample, and then it's ready for analysis. Certain machines, certain instruments can handle highly acidic acid like this. Okay. If we opt for combustion, dissolution of ash, we are also preparing sample in acidic form. So there are machines that can take this, and there are machines that can handle high matrix interference like liquefaction for extraction of inorganics. Look at the instrument, but before that, this is another example of sample that we can liquefy in order to get liquid. Okay, I have uploaded the video at Open Learning site. Okay, please access the site to look at how this instrument called sample digester looks like. It operates basically at lower temperature in comparison to muffle furnace 575. Liquefaction works at 250 Celsius degrees, which is lower. Of course, the organics, instead of converting to carbon dioxide, we get organics dissolving into the acidic solution. So you need an instrumental technique that can handle high matrix interference to get data or quantify the tin in your sample which has been liquefied. Okay, so this reaction refers to concept 1 or combustion reaction. You get mineral oxide. You can also do this acid dissolution, get prepare sample with least or less interference and run analysis. Having said that, so there are two ways here of extracting out inorganics from your bio samples. Okay, one is combustion, and then proceeded with followed by dissolution, and the other one is liquefaction. Okay, so the videos are at the Open Learning site. Feel free to access them. So I have to congratulate you. By now, you have got insight into inorganic analyte extraction from biosynthetic. See you in the next video.